Hi everyone, my name is ANGELYN Lee Jie. My metric number is 205205. Today I am going to conduct experiments on the preparation of bacterial smear and gram staining for the hands-on assessment of BMY 3201. The materials and apparatus used in the preparation of bacterial smears are glass slides, Bunsen burner, inoculating loop, and water. First, sterilize the inoculating loop by flaming it over the Bunsen burner flame and cool it for about 10 seconds. Next, place a loop full of water on the slide. Flame the neck of the tube before recapping the tube. Transfer a small amount of bacterial inoculum from the plate into the drop of water and spread both of them into a thin area. After that, allow the smear to air dry completely. Finally, heat fix it by quickly passing the smear several times over the flame of Bunsen burner. The materials and apparatus used are bacterial smear, Bunsen burner, staining tray, crystal violet, grams iodine, 95% ethyl alcohol, and safranin. First, gently stain the smear with crystal violet for 1 minute and wash off the stain with tap water. Next, apply grams iodine for 1 minute and wash off the grams iodine with tap water. Add 95% ethyl alcohol drop by drop until the alcohol runs almost clear, and wash it off with tap water. Counter stain the smear with safranin for 45 seconds and wash off the safranin with tap water. Finally, blot dry the glass slide with bibulous paper and examine under the oil immersion. Under the microscope, gram-negative bacteria will stain pink while gram-positive bacteria will stain purple. The differential nature of the gram stain is based on the ability of the bacterial cells to retain a primary stain by resisting the decolorization process. Gram staining involves 4 chemical reagents and 4 important steps. First, the cells are stained with crystal violet, followed by the addition of a mordant, which is the grams iodine, to sharpen the color of the primary stain. Next, the decolorizing agent, 95% ethyl alcohol, is applied to selectively remove the stain from the gram-negative bacteria. During the decolorization, if the primary stain is not washed out, the counter stain cannot be absorbed and the cell or its components will retain the color of the primary stain. If the primary stain is removed, the decolorized cellular components will accept the contrasting color of the counter stain. Finally, a secondary stain, safranin, is added, which counterstains the decolorized cells pink. Gram staining differentiates and divides bacterial cells into 2 major groups, which are gram-positive and gram-negative bacteria. The gram stain reaction is based on a difference in the chemical composition of bacterial cell walls. Gram-positive cells have a thick peptidoglycan layer, while gram-negative cells have a thinner peptidoglycan layer and surrounded by outer lipid layers. Therefore, gram-negative cells will absorb the counterstain which stain pink, while the gram-positive cells will retain the purple color of the primary stain.